Now next lecture is the organization of eukaryotic DNA, how a very long molecule in case of eukaryotes they form a compaction and can be organized into a very small organelle that is in the nucleus. The human cell it contains the 46 chromosomes and total DNA is approximately 1 meter long. It means each chromosome it contains 1 meter long DNA. And the genetic material is effectively packing or packed into a cell nucleus so that it can be efficiently replicate and its genetic information can efficiently be expressed. So a very long molecule, it not only be replicated but also can express its genetic information, and also it show a compaction within the nucleus. This compaction it requires the interaction of DNA with a large number of proteins, and this interaction it specifically it function in the order packaging of these long molecules of DNA. The eukaryotic DNA is associated with a tightly bound basic proteins which is known as the histone. Histones, these are the basic proteins by which they are associated with the DNA, and these histones they form the compaction of DNA into the nucleus. These histones they form the fundamental structure units which is known as the nucleosome, and further arranged into the uh, more complex structure that organize and then condense the long DNA molecule into a chromosomes, and these chromosomes they can be segregated during a cell division. Now what happened or how these histones they interact with the DNA so as to form the nucleosomes. Nucleosomes it is the structural units of the chromosome or chromatid. There are the 5 classes of the histones which are designated as H1, H2A, H2B, H3 and H4. These 5 classes of histones they interact with the DNA so as to form the compaction. These histones they are the basic proteins. They contain the high content of the lysine as well as the arginine residues, and due to the presence of these basic amino acids they are positively charged, while the phosphate groups present on the DNA which are the negatively charged. These positive charge and the negatively charged phosphate group they interact to each other and they form the nucleosome. Histones along with the positively charged ions such as the magnesium also they help to neutralize the negatively charged DNA phosphate group. This is the formation of the nucleosome. Here the nucleosomes they form the core H2A, H2B, H3 and H4, while the H1 it is present, this histone is present in the linker DNA. These histones they interact with the DNA through the interaction of the lysine or the positive charge which is present on the lysine or arginine basic amino acids and the negatively charged phosphate group. These nucleosomes they form the compaction of the very long molecule of DNA. The N-terminal ends of these histones they can also be acetylated, methylated or phosphorylated, and these covalent modification they also influence the tightly bound histones with the DNA, as during the gene expression the acetylation take place. This acetylation it diminish the positive charge of the basic amino acids. So the loose interaction between the DNA and the histone molecules, and it also affect on its conformational changes. This is known as the DNA remodeling or chromatin remodeling. So this happened during the expression of a specific genes. The neighboring nucleosomes, they joined by a linker DNA approximately 50 base pairs long. Histone H1. Histone H1, it is present in the linker DNA. And it also facilitated the packing of the nucleosomes into the more compact structure. This is the DNA. Here, compaction takes place by the formation of the nucleosome, while the H1 histone, it is present on the linker DNA. More compaction like here, and similarly more and more DNA molecule it wound, it packed, or it is coiled, super coiled. This one is the scaffold protein, scaffolding of the nucleosomes. So nucleofilament, it is coiled and anchored to scaffold protein, around which the nucleofilament it is coiled. By this arrangement, the DNA molecule it is more and more packaged into the nucleus. The polynucleosomes, they form the nucleofilament, and they assume the shape of a coil, often referred to as the 30 nanometer fiber, and these fibers now organized into a loops anchored by a nuclear scaffold containing several proteins, and additional level of organization lead to the final chromosomal structures. This one is another representation here, the DNA, then the formation of the nucleosome, then the a chromatin, that is the 30 nanometer about width is formed, more coiling take place, again more coiling, and finally the 1400 nanometer thick chromosome is formed. Now how this is the arrangement, that how DNA it is the organized into a chromosome.